Hi students, that in previous video we come across what actually the process of amino acylation or charging of amino acid. Okay, ನಾವು ಅಲ್ಲಿ ಏನು ಕಲ್ತ್ವಿ, ಹೇಗೆ ಒಂದು ಅಮೈನೋ ಆಸಿಡ್ನ ಚಾರ್ಜ್ ಮಾಡಿ ಆ ಅಮೈನೋ ಆಸಿಡ್ನ ನಾವು tRNAಗೆ ಬೈಂಡ್ ಮಾಡಬೇಕು. ಆಮೇಲೆ this tRNA is going to act like the amino acyl tRNA ಆಗಿ that tRNA is going to help in case of identification of codon ಅಂತ ಹೇಳಿ ನಾವು ನೋಡಿದ್ವಿ. Now we are going to study the next step of the translation, that is initiation. The process of initiation is what you call one of the important step in case of you can say in a translation. So why? Yakappa andre here the initiation is specifically considered as the formation of mRNA and ribosome complex. ಇದನ್ನ ಕಾಂಪ್ಲೆಕ್ಸ್ ಅನ್ಬಹುದು ಅಥವಾ ಬೈಂಡಿಂಗ್ ಆಫ್ mRNA ಟು ದಿ what you call ribosome ಅಂತ ಕೂಡ ನಾವಿಲ್ಲಿ ಹೇಳ್ಬೋದು. Okay, so you can say the binding of ribosomes to mRNA. Okay, so mRNA is going to bind with the what you call ribosome, that is also we can call as a initiation. So how this process is going to take place and what kind of factors are going to involve, that what we are going to study here. Okay, so here also we are going to get a very slight difference between the what we can say the prokaryotic and eukaryotic cells. Okay, first important step is the identification of specific sequence of mRNA and binding of the smaller subunit of what called ribosome to the mRNA. Okay, so nimagella gottu we have two different kind of ribosomes on the basis of their presence in eukaryotic and prokaryotic cell. Hauda nimagella gottide. So what kind of ribosomes we have? We have 70S and 30S kind of ribosome. Okay, so when you are going to look at the prokaryotic cell, we are going to get 70S types of or type of ribosome. Okay, so 70S type of ribosome is made up of two subunit, that is 50S larger subunit and 30S smaller subunit. Okay, so nimagella gottide because you already studied this in your first year syllabus. Okay, and in case of eukaryotes we are going to get 80S ribosomes. Howda, we are going to get 80S ribosomes. 80S ribosomes hege ಮಾಡಿರ್ತದಪ್ಪ, that is actually the larger subunit is 60S and smaller subunit is 40S. Okay, 60S ಮತ್ತೆ 40S large ಮತ್ತೆ small subunit ಇಂದ ನಮಗೆ eukaryotic ribosome ಆಗಿರ್ತದೆ. Okay, and also the mRNA that is common for both the prokaryotic and eukaryotic after what you called the process of splicing in case of eukaryote. Okay, so in the mRNA we are going to get the 5' cap which is made up of the what you called methylated or methylated G cap anta kuda adanna karitare. Okay, so here a very what you called uneven kind of the G or the guanosine is going to attach to the or present at the beginning of the mRNA, and end is having many A's, that is what we call 3' end is called poly A tail ಅಥವಾ 3' end of the what you call mRNA ಅಂತ ನಾವಿಲ್ಲಿ ಕರಿಬಹುದು. So now we are going to get the sequence on this the particular mRNA. Okay, so I am repeating this because it may help you to recall the actually what we studied in previous what you called the video. Okay, so what we are going to get here, that is UTR region as well as the specific what we called mRNA sequence, and that is what actually we are going to get AUG, the start codon, and after that GAA or GUA or GAU, a stop codon, any one of this, and again UTR region. Okay, so idu namage mRNA siktade. Okay, so what is actually initiation? That is formation of mRNA and ribosome complex. Hauda, binding of ribosome to the mRNA anta na helide. So to do this we need energy, and also we need what we called a specific factors. Okay, so that is what we called initiation factors. Okay, so what is that? Initiation factor. To do this we need what? The initiation factors. Now adanna IF anta baritini. Okay, so we are going to get two different kind of initiation factors with respect to the two different kind of cells. One is in case of prokaryote, and second one in case of eukaryote. So prokaryotically we are going to get three different kind of initiation factors, that is initiation factor 3, initiation factor 2 and initiation factor 1. Okay, there are three different kind of initiation factor we are going to get that are going to involve in case of the process of the formation of the mRNA and the ribosome complex. Okay, and in case of eukaryote we have totally 9 varieties of initiation factor, that is I am going to represent as EIF. Okay, initiation factor 1, initiation factor 2, initiation factor 1, initiation factor 2, initiation factor 3 and initiation factor 4A, 4B, 4C, 4D, 5 and 6. Okay, there are 9. How many? 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are what we have 9 different kind of initiation factors in case of what? The eukaryotes. Okay, so prokaryotically 3 ide, eukaryotically total 9 ide. So all are going to do a different kind of work, or they have a specific what you call function during the process of initiation. So ಕೆಲವೊಂದು premature subunit binding na break ಮಾಡಿದ್ರೆ ಅಥವಾ stop ಮಾಡಿದ್ರೆ, some are going to help in case of recognition of the mRNA sequence, and some of them are going to help in case of identification of the start codon, and some are going to help in case of the process of holding. Okay, so holding the smaller and larger subunit after initiation or at the time of tRNA binding to the mRNA. So they have a specific function in the what you called the specific time of or at the time of the initiation. Okay, so they have in case of eukaryotic we have 9 initiation factor, in case of prokaryotic they have only 3 initiation factors. With this we require the very important thing, that is Mg+2 ions. Okay, so namge ಏನು ಬೇಕು? Mg+2 ions. And we know that amino acids are already charged. So amino acid charge ಆಗ್ಲಿಕ್ಕೆ ನಮಗೆ ATP use ಮಾಡ್ಕೊಂಡ್ಬಿಟ್ಟು they are going to charge, and we are going to get the charged amino acid. So to bind this ribosome to that of the mRNA we need again energy. That energy is actually not ATP here, instead of that it is GTP. ನಮಗೆ ಏನು ಬೇಕು ಇಲ್ಲಿ? GTP energy na ನಾವು ಇಲ್ಲಿ use ಮಾಡ್ಕೋಬೇಕು, or cells are going to use the GTP as the next energy source which are involving in case of the process of translation. So ee energy yaak bekappa, that is we are going to study in what you called upcoming videos. Okay, so what we need here? The factors. What are those factors? Initiating factors. Initiating factors are in prokaryotic are 3, and in case of eukaryotic they are 9. Okay, so with this what we require? With this we need the what we called energy, that is energy in the form of GTP, and also we need the one more important what we call component, that is what we can call the Mg+2. Okay, these are the requirement of what you called the factors for the formation of complex or assembly of assembly of mRNA to that of what we call the ribosomes. Okay, so in case of prokaryote as well as in case of eukaryote. Okay, so hope you understood what is actually this. Now, how actually initiation takes us? Let's study. Okay, so I am going to rub this. Here, the initiation takes place by binding of smaller subunit of ribosome to the mRNA. That is the first and most important process of initiation. So what happens? First, the smaller subunit of ribosome is going to bind to the mRNA at the site where the start codon is present. Okay. So, in case of prokaryote, this process is going to be what we called initiated by the initiation factor 1 and 2. In case of eukaryote, it is going to be initiated by the initiation factor what we called 1, 2, 3 and the initiation factor 4s. Okay. So, different are there. Okay. So, bere work martavu adanna illi namge avashyakate illa. Okay. So, that is initiation factors are going to help in case of identification of the start codon and what we call that is a binding of smaller subunit to the mRNA what we called unit. Okay. So, where there is a start codon, and in case of prokaryotic specific sequences are there in case of mRNA. That sequence are going to help in case of identification of the beginning or cap region of mRNA and binding of the particular the ribosome smaller subunit. So, this smaller subunit prokaryotically enagirtadappa, this smaller subunit in case of prokaryote is 30s, in case of eukaryote it will be 40s. Got it? So, adu one difference agi navilli nodbodu. So, that is smaller subunit which is going to bind during the initiation process of what we called the translation. The prokaryotic will be 30s and eukaryotic will be 40s. So, where they are going to bind? Where there is a start codon, alli adu bind agtadu. And second thing, now other initiation factors are going to involve in case of bringing or directing the tRNA which is charged and initiating tRNA, especially they are going to what we called the guide the initiation tRNA. So, in case of initiation tRNA, prokaryotic is the tRNA which is having the F met. In case of eukaryote, it is just the methionine. Artha. So, in case of prokaryotically, adu formaldehyde methionine agirtade. In case of eukaryote, adu just methionine agirtade. Okay. So, now what happens? In the presence of Mg plus 2 ions and with the help of GTP, okay, energy rich molecule GTP, the large subunit going to bind the mRNA, okay, mRNA. So, before this formation of the larger and smaller subunit complex, idarkinta munche, there is attachment of tRNA to the start codon. Start codon ge tRNA bind agodu one what we called step ide. Okay, that is before the attachment of what? Attachment of this larger subunit. So, when the larger subunit attaches before that, okay, so we can see the identification of the start codon by the tRNA, okay, that is F met in case of prokaryotic and methionine in case of eukaryotic. Okay, so I am going to write here F met only, so we will take only prokaryotic first. Okay, so illen agtada adu bind agtada, and later with the help of GTP and Mg plus ion and different kind of what we call initiation factor, that is larger subunit is going to bind to the mRNA. Now the complex is complete. Yenitappa actually initiation andre that is binding of ribosome to the mRNA. So ಏನಾಯ್ತಿಲ್ಲಿ, so smaller subunit and larger subunit with a charged initiating tRNA is going to form a complex that is done here. But very interesting thing is that the larger subunit is not like a smaller subunit. Why? Because larger subunit is divided into 3 different chambers. Okay, larger subunit alli now 3 different chambers na nodabodu. So these 3 different chambers are site A, site P and site E. Okay, site A is commonly called as amino acyl site. Okay, idina amino acyl site anta karitare. P is the site where actually, sorry, first amino acyl site ಬಗ್ಗೆ ತಿಳ್ಕೊಳ್ಳೋಣ. Amino acyl site is a site where the amino acid containing tRNA are going to enter first, okay, to their respective what you called codons. So ಏನು ಮಾಡ್ತವೆ, charged amino acid na ತಗೊಂಬಂದಿರೋ tRNA ಏನು ಮಾಡ್ತವೆ, they are going to enter the A site, okay, and they are going to form a bond between the codon and anticodon. ಅದು ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಆಗಿ ನಾವಿಲ್ಲಿ ನೋಡಬಹುದು. ಓಕೆ, ಸೊ ಇದಾದ್ಮೇಲೆ this second step or second what we call chamber or second site what we have, that is P site. P site is specifically made for formation of peptide bond. Okay, idenappa idu made for formation of the peptide bond between the incoming amino acid and pre-existing amino acid of P site. So the P site is called peptidyl site anta karitare. P site is called peptidyl site. So the site where the formation of what we call the bond between 2 amino acid, one is already present in the P site, one is just entering the A site, just enter the A site. ಅವು ಎರಡರ ನಡುವೆ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಆಗುವಂತ ಒಂದು ಸೈಟ್ ಏನಪ್ಪಾ, ಅದು P site. That's why ಇಲ್ಲಿ ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್ ಆಗೋದ್ರಿಂದ ಅದನ್ನ ಪೆಪ್ಟೈಡಲ್ ಸೈಟ್ ಅಂತ ಕರೀತಾರೆ. So after this we are going to get site E. What is site E? Site E, when the P site tRNA is going to form bond between the A site amino acid, okay, is now going to lose its amino acid. Okay, adenagtada tana amino acid na kalkonbiಡ್ತದೆ. So now this tRNA is going to reach the site E and going to enter out of the what, the box or out of the ribosome. ಈ ribosome ಇಂದ ಹೊರಗಡೆ ಹೋಗ್ತದೆ. So this site is specially for exit of tRNA. That's what is called exit site anta karitave. That is what we call tRNA exit site. Okay, so what we have 3 chambers. First chamber is A site, second is P site and third is E site. Okay, so ಇದನ್ನ ನಾವು ಈಸಿಯಾಗಿ ನೆನಪು ಮಾಡ್ಕೋಬೇಕಾದ್ರೆ ಏಪ್ ಅಂತ ನೆನಪಿಟ್ಕೊಳ್ಳಿ. APE, ape ಅಂದ್ರೆ monkey. Okay, so ape APE. A is for amino acyl site, P for peptidyl site and E for tRNA exit site. This is what initiation. So tRNA, the first initiation codon identifying tRNA which is carrying methionine is going to enter P site, not A site. That is very interesting thing. Okay, so in case of this process, that is in case of initiation, the first tRNA athwa amino acyl tRNA is going to enter or attach, okay, attaches to the P site of larger ribosome subunit. Okay, ಯಾವ subunit ಅಲ್ಲಿ larger subunit ಅಲ್ಲಿ P site ಅಲ್ಲಿ ಇರೋ initiation codon ಇದು identify ಮಾಡುತ್ತೆ. So here we are going to get the UTR, untranscribed region ಬಿಟ್ಬಿಡಿ, ಅಲ್ಲಿ ಏನು ಇರಲ್ಲ. Okay, so the P site which is having AUG is going to identified by the initiation amino acid, that is F-met amino acid, and going to bind to the P site only. The first amino acid or amino acid RNA L bind agtadappa, that is in a P site. Okay, so this is regarding what the initiation. Okay, so initiation of what the what you called the amino acid chain synthesis athwa peptide chain synthesis. This is initiation process. Okay, so let's study the second one, that is what we call the elongation. Okay. Okay, so that what we studied, the process of initiation. So now we'll study the process of elongation. So elongation is nothing but the formation of polypeptide chain. Okay, so polypeptide chain formation ನಾವು elongation ಅಂತ ಕರೀಬಹುದು. Okay, so you know very well that at the stage of initiation, so that what we have, that is because of the what we called the different factors called elongation, that is initiation factors in case of prokaryotic and initiation factor in case of eukaryotic. Okay, so E stands for eukaryotic initiation factor and IF initiation factor. Okay, so in case of prokaryotic, so they are going to help in case of formation of what? The formation of the complex of mRNA and ribosome. Idu ready aaytu. So now next step is addition of amino acids continuously to that of the initiated what you call it the chain elongation process. Okay, so initiation ಆಯಿತು, ಈಗ chain elongation ಆಗ್ತದೆ. So in this step what is going to happen? So that in case of cytoplasma, this is the process which is taking place in the cytoplasma. In cytoplasma we have a large number of specific tRNA which are carrying what we called their respective amino acids. Okay, so ಇಲ್ಲಿ ಸಾಕಷ್ಟು tRNA ಇರ್ತಾವ, they are going to carry their own amino acids, so which are they are going to code for, okay, specifically. So ಸಾಕಷ್ಟು amino acids ನಾವಿಲ್ಲಿ ನೋಡಬಹುದು, so which are carried by the tRNA. So now according to their initiation and according to their what we call codon which is present on the mRNA and the anticodon which is present on the tRNA, they will come and bind to the what we called mRNA at A site. Okay, so enagtavu now because of factors which are present in the cell cytoplasma, this tRNAs which are having a specific amino acid are able to bind to the mRNA with the help of what we called the factors. Those factors are called the elongation factors. They are called elongation factors. Elongation factors in case of prokaryotic and elongation factors in case of eukaryotic are different. So ಬೇರೆ ಬೇರೆ elongation factors ನಾವು ನೋಡಬಹುದು. Usually in case of eukaryotic we are going to get the number, in case of prokaryotic we are going to get the alphabets. Okay, so different alphabet G they what you call like that different alphabets we are going to get in case of the prokaryotic. So we have different kind of prokaryotic elongation factors as well as eukaryotic elongation factors. This prokaryotic in case of prokaryotic cell and eukaryotic in case of eukaryotic cell are going to help in what you called identification of specific site and especially A site of the tRNA. So what happens now? The next tRNA which is entering, okay, so to the A site with respect to its specific what you called amino acid sequence is present here. Just consider it is charged, okay, amino acid is charged, or consider this one is leucine, okay, leucine anta tilkodri. Now this amino acid is going to enter and form a bond between the codon and anticodon. Okay, so what is going to happen? Here in case of mRNA we have codons, and in case of tRNA we are going to get anticodons. Okay, so codon anticodon eradu anti parallel irodrinda and complementary irodrinda they are going to form a bond here, and here we are going to get amino acid. So that is leucine anta tilkolli. Okay, so this is first one, this is second amino acid. Now in P site, okay, so A site is for entry of what? Amino acyl tRNA, and in P site there is a reaction between the first amino acid and second amino acid to form a bond called the first formulated methionine is going to bind to the next leucine or whatever amino acid by the help of peptide bond. Okay, by the help of peptide bond. So first amino acid is going to react with the second amino acid to form the peptide bond. For this there is an enzyme in this particular chamber, okay, or associated with this particular P site. That associated enzyme is peptidyl synthase or peptidyl transferase. Okay, so that enzyme is peptidyl synthase or peptidyl transferase anno enzyme ಏನು ಮಾಡ್ತದಪ್ಪ, it is going to form a bond between methionine of the first amino acid or the first amino acid to that of the second amino acid. It is going to form the peptidyl bond. And now this T or tRNA present in case of P site is now going to move to the next T site. Okay, this T site ಗೆ ಬಂದು this tRNA now it is going to be released out by separating from its amino acid. ಏನಾಗ್ತದಪ್ಪ ಇಲ್ಲಿ, it is going to separate from its amino acid and it is sent to the next one, and now again the A site is free. ಏನಾಗ್ತದೆ ಇಲ್ಲಿ, that is I am going to make a small model here to show you that. Okay, ಏನಿತ್ತು ಇಲ್ಲಿ, that is it is present in case of A site, P site and T site is empty. So P site alli namage first amino acid ide, and in case of second, that is in a A site we have what we called second amino acid containing tRNA. Now the bond is going to transfer. Okay, so A is going to bind with a what we called the first amino acid is going to bind with the second amino acid, and third what we called second amino acid is now going to move to the A site. Artha aita higittu. Now this particular what we called that is ribosome is going to move to the next. Due to that, the second amino acid containing tRNA will reach the P site, and the tRNA which is donated its amino acid now going to reach the T site, what we call the E site. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ P ಅಲ್ಲಿರೋದು E ಗೆ ಬರ್ತದೆ, A ಅಲ್ಲಿರೋದು P ಗೆ ಬರ್ತದೆ. Now we have two amino acids which are formed a bond between them, that is what we call peptide bond. Now next what happens, because of the enzyme, okay, because of the enzyme called translocase, okay, enzyme called translocase, this particular what we called the ribosome complex going to move next, so that P site tRNA, okay, ಇಲ್ಲಿ ತಿಳ್ಕೊಳ್ರಿ, move next, ಅದನ್ನ next move ಆಗ್ಲಿಕ್ಕೆ translocase help ಮಾಡ್ತದೆ. Now A site is empty. Again a new tRNA is going to accumulate here with a new amino acid, and it is going to form a bond between the amino acid which is already present in the P site, and after that this translocate is going to push the tRNA next. Again the tRNA present in the P is going to reach the E. Okay, so P alliro ಏನಪ್ಪ ಇದು ಅಮಿನೋ ಆಸಿಡ್ ಟಿ ಅಲ್ಲಿ ಬರ್ತದ, ಸಾರಿ ಟಿ ಆರ್ ಎನ್ ಎ ಟಿ ಇ ಅಲ್ಲಿ ಬರ್ತದ. ಇ ಬಂದ ತಕ್ಷಣ ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಎಂಟರ್ ಔಟ್ಸೈಡ್. ಅಗೇನ್ ಎ ಈಸ್ ಫ್ರೀ. ಅಗೇನ್ ದಟ್ ಈಸ್ ಕನ್ಸಿಡರ್ ಥರ್ಡ್ ಅಮಿನೋ ಆಸಿಡ್. ಅಗೇನ್ ದ ಫೋರ್ತ್ ಅಮಿನೋ ಆಸಿಡ್ ಕಂಟೈನಿಂಗ್ ಟಿ ಆರ್ ಎನ್ ಎ. ಇನ್ನೇನಾಗ್ತದೆ ಇದು, one more tRNA will enter here, and here now we have three what we called amino acid which are made up of polypeptide bond or peptide bond, and now the third amino acid which is entering inside, athwa fourth amino acid which is entering inside, it is going to enter the A site, and the P site idu T site ಗೆ ಬಂತು, E site ಗೆ ಬಂತು, E site ಬಂದು ಹೊರಗಡೆ ಹೋಯಿತು again. So like this formation of the bond takes place. The same way this ribosome move from the 5' end till the 3' end until it is going to reach this stop codon. Enadappa illi we have stop codon. When it is going to reach this stop codon, any one of this stop codon, okay, ಯಾವುದು ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ರಿ. So just consider GAA, okay, ಯಾವುದೇ ಒಂದು stop codon ಇದೆ ಅಂತ ತಿಳ್ಕೊಳ್ರಿ. ಅಲ್ಲಿವರೆಗೆ this tRNA are going to enter and they are going to form the what we call chain. So end ಗೆ reach ಆದಾಗ this amino acid chain will be longer and it is going to form a long polypeptide chain. Estili sequence itto, how many what we called the codons are going to be carried by the mRNA, that amount of what we call the amino acids are going to join and going to form a long chain. ಅದಕ್ಕೆ ಏನಂತಾರೆ, polypeptide ಅಂತ ಕರೀತಾರೆ. polypeptide. ಇದು ಎಲ್ಲಿವರೆಗೆ ಆಗ್ತದಪ್ಪ, until it is going to reach this stop codon. So now consider this is going to reach the stop codon. This one is a what you call last amino acid which is going to bind to the it is entering in the P site with a tRNA. Now we reach the stop codon. A site ಗೆ ಏನು ಬಂತಪ್ಪ, ಈಗ ಸ್ಟಾಪ್ ಕೋಡಾನ್ ಬಂತು ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಈ ಸ್ಟಾಪ್ ಕೋಡಾನ್ ಬಂದ ತಕ್ಷಣ, ಓಕೆ, stop codon is not going to code for any kind of amino acid. We know very well that. So now what is going to accumulate here, that is releasing factors. Okay, so now releasing factors are going to act on the ribosome complex, and one releasing factor is going to attach to the A site and they are going to dissociate. Illi enagtada, they are going to dissociate. Enagtade illi, this is going to act on P site tRNA, and the tRNA now going to reach the E site and going to get released. Okay, release ಆಗ್ತದ, last tRNA is going to get released, and this releasing factors, okay, ಅಥವಾ dissociation of ribosome complex. Okay, ಏನಾಗ್ತದೆ ಇಲ್ಲಿ, dissociation of ribosome complex ಆಗ್ತದ because of releasing factors. So eukaryotic releasing factors ಇದಾವೆ, ಆಮೇಲೆ prokaryotic releasing factors ಇದಾವೆ. ಸೊ ಇದೆಲ್ಲ ಆಗ್ಲಿಕ್ಕೆ ಅದೇನ್ ಬೇಕಪ್ಪ, GTP ಎನರ್ಜಿನ ಯೂಸ್ ಮಾಡ್ತಿಲ್ಲ. ಏನಾಗ್ಲಿಕ್ಕೆ, that is formation of the polypeptide. When it is going to reach the last what we called the stop codon, there is nothing to form what we called the protein or what we call to bring the amino acid. That's why the releasing factor is going to bind and dissociate the complex. ಏನ್ ಮಾಡ್ತದಪ್ಪ, this complex is going to dissociate larger subunit and smaller subunit. ಎರಡನ್ನು ಬೇರೆ ಮಾಡಿಬಿಡ್ತದೆ. This is what we call termination. Okay, so the last process is what, that is what we called, I am going to explain once again. So ಇಲ್ಲಿ process of what we called the tRNA transfer ಆಯಿತು. ಸೊ ಇದನ್ನ ನಾವು ಇಲ್ಲಿ ತಗೊಳ್ಳೋಣ ಹೀಗೆ. Okay, so that we don't have anything here. So last, it is going to reach the last. Okay, I am going to explain it again. So last, what is that? There is no tRNA. Okay, last one is a termination. Ellivarge elongation factor agtada until it is going to reach the stop codon. So now consider, that is our ribosome is reached. Okay, so the A site of ribosome reached the stop codon. Now we have here one tRNA with a large chain of amino acid. Okay, on the large chain of amino acid illi anta tilkoli. So this is the site. Okay, so what we have here, the ribosome now reached the last. Okay, so here we reach the what we call stop codon. ಇಲ್ಲಿ ಕೆಳಗಡೆ ನಮಗೆ ಸ್ಮಾಲರ್ ಸಬ್ ಯೂನಿಟ್ ಇರ್ತದ. ಓಕೆ, ಸೊ ರೀಚ್ ಆಯ್ತು ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಸೊ ರೀಚ್ ಆದ ತಕ್ಷಣ ಏನಾಗ್ತದಪ್ಪ ಇಲ್ಲಿ, there is a attachment of what? Releasing factor. So ಏನಾಗ್ತದೆ ಇಲ್ಲಿ termination ಅಲ್ಲಿ there is a attachment of terminating factor or releasing factors are going to reach, and these releasing factors are going to dissociate. Okay, they are going to dissociate the complex. So they are going to help in separating the protein chain from the tRNA. Now tRNA will get released as what we called free, what we called tRNA, and the amino acid is going to, amino acid chain is going to release as a free amino acid, and tRNA will come out, and the dissociating factor or the releasing factors are going to separate the larger subunit and the smaller subunit. So this is the what? The end of the process. So end of the process enagtada, that is releasing factor, and especially by the guidance of the stop codon. There is no what we call stop codon for this GAA, GUA and GAU. So there is no any kind of coding for amino acid. So now this amino acid chain is going to be released, okay, and tRNA will get free, and with that these releasing factors are going to dissociate the larger and smaller subunit chain. The complex is now going to be dissociated. Yakappa antandre, now the mRNA is going to produce the complete structure of protein. Namak ಬೇಕಾಗಿರುವಂತ protein mRNA ಈಗ produce ಮಾಡಿಬಿಟ್ಟಿದೆ. Okay, so that's why the releasing factors are going to release the all thing that which are involving in case of the process. Okay, that is what we call termination. Okay, so elongation ಆದ್ಮೇಲೆ ಬರೋದು ಯಾವುದು? Termination. So termination ಅಲ್ಲಿ, okay, so ನಾವು ಇನ್ನು ಸ್ವಲ್ಪ ನೋಡಬೇಕಾದ್ರೆ, so when the termination is going to take place in case of prokaryote, okay, in case of prokaryote, we are going to get the protein chain, okay, made up of amino acid, but the first protein or first amino acid of the protein is F-methionine. In case of eukaryotic, that is ulta. Okay, it is not having anything else than that of the amino acid. This is completely formed primary protein. Artha aita? So in case of prokaryotically methionine irodrinda, that is what we called one more process. Okay, so it is going to be seen in case of that is in a prokaryote, that is what we call the modification. Okay, ಇಲ್ಲಿ ಏನಾಗ್ತದೆ? Modification ಆಗ್ತದೆ. Modification. So modification ಅಂದ್ರೆ ಏನಪ್ಪ ಇಲ್ಲಿ? Just demethylation, sorry, deformylation. Okay, deformylation of polypeptide ಅಥವಾ ಸ್ಪೆಷಲಿ ಮಿಥಿಯೋನೈನ್ ಅಂತಾನೂ ಕರಿಬಹುದು. ಸೊ deformylation of polypeptide ಆಗುತ್ತೆ. That is done by what we called the enzyme called deformylase. Okay, deformylase ಅನ್ನೋ enzyme ಏನು ಮಾಡ್ತದೆ? Modification ಮಾಡ್ತದೆ. That is fifth step in case of what? Prokaryote. Okay, so prokaryotically fifth step anta ನಾವು ಅದನ್ನ ಕರಿಬಹುದು. But in case of eukaryote, we are going to get the end, what we called the completely what we called free or completely formed a sequence of amino acid without any kind of the problem or modification. Okay, it does not require any kind of modifications. And one more thing what we have to concentrate here, that is very interesting thing. Okay, so the one more thing that what we have to concentrate here, that is formation of polyribosome. Okay, so very interesting. ಸ್ವಲ್ಪ ಕೇಳ್ರಿ ಇದನ್ನ, formation or polyribosome ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಸೊ ಇದನ್ನು ನೀವು ಎಲ್ಲಾದರೂ ಕಲ್ತಿದ್ರೆ ನೆನಪು ಮಾಡ್ಕೊಳ್ಳಿ. ಸೊ ಡೆಫಿನೆಟ್ಲಿ ಇದನ್ನು ಕಲ್ತಿರ್ತೀರಾ ಇನ್ ಕೇಸ್ ಆಫ್ ವಾಟ್ ಯು ಕಾಲ್ಡ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಸೆಲ್ ಅಲ್ಲಿ. ಸೊ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಪಾಲಿರೈಬೋಸೋಮ್. ಏನಿದು ಪಾಲಿರೈಬೋಸೋಮ್? ದಿಸ್ ಈಸ್ ಅವರ್ mRNA which is having 5' and 3' end. So I have a very simple question for you. So is there only one mRNA which is produced by the what you call the specific structural gene at particular time, and is there only one ribosome is going to produce only one protein at the from that particular mRNA? Okay, so simple question ಇದೆ. ನಮಗೆ ಸ್ಟ್ರಕ್ಚರಲ್ ಜೀನ್ ಇಂದ ಒಂದೇ mRNA ಬರ್ತದ? ಫಸ್ಟ್ ಕ್ವಶನ್. ಸೆಕೆಂಡ್ ಒನ್, ಒಂದು mRNA ಇಂದ ನಮಗೆ ಒಂದೇ ಒಂದು ಪ್ರೋಟೀನ್ ಚೈನ್ ಸಿಗ್ತದ ಅಥವಾ ಆ ಸೇಮ್ ಪ್ರೋಟೀನ್ ನಾವು ಬಹಳಷ್ಟು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು ಅಥವಾ ಮಾಡ್ತಿರ್ತದೋ ಸೆಲ್? ದಟ್ ಇಸ್ ಅ ಕ್ವಶನ್. ಎಸ್, ದಟ್ ಈಸ್ when you are going to look at the mRNA, we are going to get the many ribosome attached to the one single strand of single strand of the mRNA. Okay, so ಇಲ್ಲಿ ನಾವು ಅನ್ಕೊಂಡಿರೋ ಹಾಗೆ ಒಂದು ರೈಬೋಸೋಮ್ ಒಂದು mRNA ಮೇಲೆ ರನ್ ಆಗಿ ಒಂದೇ ಒಂದು ಪ್ರೋಟೀನ್ ಚೈನ್ ರೆಡಿ ಮಾಡಲ್ಲ. So here many ribosome according to the requirement of the cell. Okay, so ನಮಗೀಗ ಎಷ್ಟು ರಿಕ್ವೈರ್ಮೆಂಟ್ ಇದೆ ಸೆಲ್ ಅಲ್ಲಿ, ಎಷ್ಟು ನಮಗೆ ಪ್ರೋಟೀನ್ ಬೇಕು ಅಷ್ಟೆಲ್ಲ ಪ್ರೋಟೀನ್ ರೀಚ್ ಆಗೋವರೆಗೆ the continuous process takes place with a multiple ribosome complex. One ribosome complex ready ಆಯ್ತಾ, it is going to run and going to end. ಒಂದು ಮುಂದೆ ಹೋದ ತಕ್ಷಣ second one. Okay, this one is a first which is released and this is going to release one protein chain. Okay, this was the second. ಅದಾದ್ಮೇಲೆ ಇದು second start ಮಾಡಿತ್ತು. This is still have to reach the stop codon and going to release the protein, and this one is a third. It is still what you called have to run a little bit longer, and this one is a fourth and is having few what you called the amino acids. This one is a fifth, it is having still more less amino acid than the fourth one. This one is a sixth, it is just initiated, that's why having only 2-3 amino acids chain. Okay, this is how the protein synthesis takes place. This is what we call polyribosome formation anta karitare. So ಒಂದೇ mRNA ಇಂದ we are going to get a lot or thousands of what we called copies of protein or the polypeptide from the one mRNA. ಸೊ ಒಂದು mRNA ಸಾಕಷ್ಟು ಪ್ರೋಟೀನ್, ಸಾಕಷ್ಟು ಸಾವಿರಾರು ಗಂಟಲೆ ಪ್ರೋಟೀನ್ನ ರೆಡಿ ಮಾಡ್ಬೋದು. ಯಾವಾಗ? when there is a running of more than one that is a ribosome complex on the mRNA strand. It is going to happen regularly in the cell. So ನಮ್ಮ ಸೆಲ್ ಅಲ್ಲಿ ನಮಗೆ ಒಂದು 100 strand of what you called 100 ಪ್ರೋಟೀನ್ ಚೈನ್ ಬೇಕಾದ್ರೆ, ಒಂದೇ ಪ್ರೋಟೀನ್ 100 ಕಾಪಿ ಬೇಕಾದ್ರೆ, ಆ 100 ಕಾಪಿ ಆಗೋವರೆಗೆ ಹೊಸ ಹೊಸ ರೈಬೋಸೋಮ್ ಬರ್ತದ, ಕಾಂಪ್ಲೆಕ್ಸ್ ಮಾಡ್ತದ, ರನ್ ಆಗ್ತದ. ಓಕೆ, ಸೊ ಯು ಕ್ಯಾನ್ ಕನ್ಸಿಡರ್ ಇಫ್ ವಿ ನೀಡ್, ಜಸ್ಟ್ ಕನ್ಸಿಡರ್ ದಿಸ್ ಒನ್ ಇಸ್ ಅ ಪ್ರೋಟೀನ್ ಎ. ಓಕೆ, ಇದು ಯಾವುದಿದು? ಪ್ರೋಟೀನ್ ಎ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ಈ ಪ್ರೋಟೀನ್ ಎ ನಮ್ಗೆ ಎಷ್ಟು ಬೇಕೀಗ ನಮ್ಮ ಸೆಲ್ಲಲ್ಲಿ? 1000 ಪ್ರೋಟೀನ್ ಎ ಕಾಂಪ್ಲೆಕ್ಸ್ ಬೇಕಪ್ಪ ನಮಗೀಗ. ಪ್ರೋಟೀನ್ ಎ ಬೇಕಾಗಿದೆ ನಮಗೀಗ ನಮ್ಮ ಸೆಲ್ಲಲ್ಲಿ. ಸೊ ಹೌ ಮೆನಿ ವಾಟ್ ಕಾಲ್ಡ್ ರೈಬೋಸೋಮ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ರನ್? ದಟ್ ಈಸ್ 1000 ರೈಬೋಸೋಮ್ಸ್. ಓಕೆ, ಸೊ 1000 ರೈಬೋಸೋಮ್ಸ್ ರನ್ ಆಗಿ 1000 ಪ್ರೋಟೀನ್ ನ ರೆಡಿ ಮಾಡ್ತವೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಇಸ್ ಹೌ. ಸೊ ಅದನ್ನ ಕೆಲವೊಂದು ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ನಾವು ನೋಡಿದಾಗ we are going to get many ribosomes on a single strand of mRNA. This structure is called polyribosome. So all ribosomes are having their respective assemble ಆಗಿರುವಂತ ನಮಗೆ amino acid na ನಮಗೆ ತೋರಿಸ್ತವೆ. Okay, so you can say the first one is having less, second one is little bit more, third one is little bit more, like this, and final one is having the completely mature what we call the polypeptide. So like this, we are going to get the many ribosomes on a same string. This is what we call polyribosome. This is what we call the end of the process called termination. Elongation means elongation of the ribosome, elongation of the polypeptide, and termination means termination of the polypeptide. Polypeptide termination agಲಿಕ್ಕೆ ಏನು ಬೇಕು ನಮಗೆ? Releasing factors. Releasing factors. So these releasing factors are present in case of the eukaryote as well as in case of prokaryote. ಈ releasing factors ಯಾವುದರ ಮೇಲೆ ವರ್ಕ್ ಮಾಡ್ತಾವಪ್ಪ? That is the stop codon. Okay, so stop codon ಮೇಲೆ ಇದು ವರ್ಕ್ ಮಾಡ್ತದೆ. That stop codon is very important for what we called identification of the end of the chain and termination of the chain, and the dissociation of the what we call complex also. So ಇದನ್ನೆಲ್ಲದಕ್ಕೆ ನಮಗೆ ಏನು ಬೇಕು? Dissociate ಆಗಲಿಕ್ಕೆ ಈ ಎಲ್ಲ ಇನಿಷಿಯೇಷನ್ ಬೇಕಾಗ್ತದೆ. Okay, so this is regarding what the process called the translation, the protein formation. So hope you got the answer that how actually the coded information present on the DNA is going to transfer to the or going to express in the cell in the form of protein anta. Okay, so now ಏನು ಕಲ್ತ್ವಿ? DNA replication, translation and transcription. Okay, so transcription and translation. Okay, so this is the process of transcription and translation as well as the process of the what we call the transcription and translation. So ella aadmele namaga last ge ಏನು ಸಿಗುತ್ತಪ್ಪ? ನಮಗೆ ಬೇಕಾಗಿರುವಂತ protein. So that what we are going to get, that what we need actually in the cell. So like this, we have a huge number of genes in our body, and every gene or structural gene is going to produce a respective mRNA. That respective mRNA is going to produce a respective what we called the protein. That all mRNA or all genes are not expressed in all the cells. Wherever it is required, they are going to express in that particular region. Okay, so for example, ನಮ್ಮ skin ಮೇಲೆ ಕೆಲವೊಂದು part ಅಲ್ಲಿ ನಮಗೆ hairs ಇದೆ, ಕೆಲವೊಂದು part ಅಲ್ಲಿ hairs ಇಲ್ಲ. Okay, so the cells which are present here, they don't require any kind of hair. That's why the hair gene or hair producing gene is not expressing here. It require here, that's what is going to express here. So like this, the cells are going to control what we call the expression of the gene. Okay, so why I am explaining this all? Because the next topic is the regulation of gene. That what we are going to study in the next concept or in the next video. Okay, thank you. So keep watching. If you have any doubt, you can ask me. Okay, so most of you not asking any kind of doubt, please try to discuss in the biology discussion group. Thank you.